handout, it's a little, this doesn't have anything to do with what I'm going to talk about, but it kind of has to do with something to kind of help you be better presenter. And I'm going to say the same sentence as Emmanuel. Not that I'm a good presenter or anything. I'm trying to be a better one. But this is a handout that Ali actually uploaded in a folder with your literature readings. And it's a cool thing to kind of glance through, because it tells you about different things that you should follow in order to make your presentation successful, both for the oral presentation, how to prepare slides, how to make them visible, what fonts to use, and also for the posters. And I find this thing really, really useful. So just kind of glance through it, and I use it as a reference point. How big should I make my my title on my poster? And it says there really specifically, this is the size of font that you should be using. So it's kind of cool. But today I'm going to be talking about proxies. So a little bit softer, a little bit lighter thematic than the one that Emmanuel was talking about. So if you think about any experiment that you have ever done, either in the lab or you went out on the ocean, you dropped down the CTD. Does everybody know what CTD is? When I say CTD, okay? That's that thing that you pass by when you go into the bathroom. So that's a small CTD, and that one has, they can measure temperature, conductivity, depth. Then it can measure fluorescence, chlorophyll fluorescence, backscattering, attenuation, has a C star on it. And then it has those 3 Niskin balls. And usually we collect with those water samples that allows us to do additional analysis. And the additional analysis can be like chemistry. So I don't know. You can do trace metals if you're crazy. You can do some additional just basic chemistry, carbons, nitrogen, like particulate nitrogen. You can do take the samples for phytoplankton community composition, so on and so on. But what it turns out to be that you always, at least it turns out to be with me, that when you look at your whole experiment, there's always a large number of parameters that have a small resolution. So, for example, I have all my biology, all my chemistry, and so on. I just collected it 3 times. Just to 3 depths. Why? Because it really takes a long time and lots of money to process those samples. But when it comes to physics, for example, temperature, salinity, and so on, I have collected lots of samples. And it's usually a small number of parameters that I can collect on high resolution. That's kind of how I see every experiment. And as I said, as it turns out to be, if you start doing biogeochemistry, which most of you guys are doing, the biogeochemical samples are sitting on this side and physics is sitting on this side. But thankfully, since optics falls into physics, we can make a connection between those 2 things, and artificially, so by some kind of correlation, I don't know, causation, whatever, we can discuss this stuff later, we can artificially increase the number of the samples of the one parameter. And that's how we develop a proxy. It's just kind of like a visual, how I see development of the proxy in some kind of weird sample parameter space. And the word proxy has its own meaning, but if you think about optical proxy, it's a measurement of a physical quantity that is optics, so any kind of optical parameter, that is used as an indicator of the value of another biogeochemical parameters. So what I'm going to be talking today about, and the title is POC and other proxies, because I assume we can't get to we can't get it to a better title, but I'm going to be talking to you about these different proxies that have been published in recent, X years, and they have been used a lot in studying biogeochemistry of the ocean. So the places where we take the optics that we have measured and try to infer some biological, chemical, geological processes with them in the ocean. Why do we want to do that? So optics, either in situ or remote sense, gives us a higher resolution data set. You have seen how many data points does an ACS produce. It does, I think, in functions like 6 or 7 hertz, which means you get 6 or 7 data points per second. Once again, if you try to go and take a discrete sample to infer something about absorption, it takes a real long time for you to process. And again, there's only so much water that you can collect. Remote sensing, it's up in the sky. It sees, the whole Earth, all the oceans once a day for case of MODIS. Traditional methods, not only they're time consuming, but they're very expensive. So last night after you guys were done, you went, take a shower, had dinner, and I stayed here and I filtered HPLC samples for you that I'm going to send for analysis. Each of these samples that collected, so half a liter water, filter in that filter, I have put in liquid nitrogen, I will send it for analysis. That analysis costs $93. for each of those data points. So just imagine now, think in your head, you will become scientist, plan your budget. How many HPLC samples do you need to take? So once again, it rolls back to the optics that is cheaper, less expensive. Sampling the parameters and scale of importance. And this rolls down to this Chang and Dickey figure, and as Mary Jane told me that this was kind of modified from Stommel's figure that he originally where he talked about this time scale continuum, where you can, on time and scales, define most of the processes that are happening in the ocean. And I'm here very phytoplankton-oriented person, so I'm going to be talking about this circle here, which talks about phytoplankton blooms and grazing, zooplankton grazing. So, different changes within the scales of the phytoplankton community. And we can see that they happen on order 1 day to 1 month, or order of 10 meters to 10 kilometers. So, for example, you have a ship, and this ship goes out to sea once a month, no? And you go and take that sample once a month, and you have your 12-month data set. You can miss the whole bloom. You can miss so much. But if you deploy a mooring, a glider, a float, or anything like that, you observe this process from remote sensing from the satellite, and you have developed biogeochemical proxies that can tell you something about the phytoplankton community, you have a better knowledge of the processes that are happening there. So you can really, with some security, say what happened. You're not going to miss the whole bloom. Increasing number of data sets in situ, which are going to mostly be talking about these in situ proxies, is going to help us validate remote sensing algorithms and also high resolution biogeochemical models. There's a modeler here. There's going to be more and more modelers who are trying to use optical data to feed in their models because they cannot survive out of 5 HPLC samples we give them. They really need high resolution data sets. High resolution data sets from biogeochemistry that operate on the same time and scales as physical. So that's where we come in. That's where we produce proxies to make a world a better place, but also to help our colleagues who are modelers so they can project that stuff, for example, for climate models. Once again, why we're doing this, optical instruments are getting smaller and more robust and more diverse. And then we can start deploying them on all these platforms. So this is a sea glider. This was deployed in North Atlantic experiment. I'm going to show you a couple of papers from here. This is Collins buoy, and I just picked that one. So because I'm not going to show any data but you saw the data from that buoy today. This is a Slocum glider. This is the one that I used during my PhD thesis. This is a Tara sailboat in which, as the manual show, there was a flow-through system, which is also high-resolution optical data set that we can use for doing cool biogeochemistry. And this is a bioargon. This is Giorgio, that's her advisor. He also went to this optics class. So these are all different platforms that produce high-resolution optical data sets that we can now translate in biogeochemistry using these proxies. One other thing, glider can go in North Atlantic in the middle of the winter. We don't know a lot about what's happening in North Atlantic in the middle of the winter. Why? Just because it's disgusting up there. You're going to puke. It's terrible. Nobody's going to go up there. But if you drop a glider can stay there for 2 months at a time. You can go there, but there's a question of sanity that goes along with it. But glider can go there and give you some information about it, and that's really, really powerful. Okay, how are we doing these? Colin said the other day, anything that causes variability in a sample is an opportunity to extract additional information from that sample. And if this variability, and you have crossed over that you have messed up something in your code, or you have forgot to zero your blanks or anything that you guys get rid of, when you guys correct all your data, and you start seeing some variability, you can start building hypotheses. And maybe there's something cool behind that variability. Maybe that is the proxy that I can use to infer blah, whatever the blah is. And so I'm going to talk about all these different blahs. So I group these. Okay, if I'm talking too fast, let me know. If that doesn't make sense, let me know, okay? There's quite a lot of these optical proxies. I just listed some of them. There's many, many, many more. And every day people are developing new ones. But I group them by quantity and quality when it comes to these basic ones. So chlorophyll, we already heard a lot about chlorophyll fluorescence. We can use absorption, and you guys start using that stuff, and you're going to have data to look at that. Not only the data collected yesterday, but it's going to be data that you guys are going to collect on the lab on Friday. I'm going to tell a little bit about that. Particulate organic carbon, phytoplankton carbon. If you go through this, you're going to see that these are all the measurements of some kind of biomass or mass or abundances or quantities in the ocean. The second group is quality. These are usually ratios or weird ways of looking into optics that tell us something about the composition, usually particulate composition. So we have particulate composition. In the manual talked about some of this. You can infer particle size. You can say something about phytoplankton composition. You can say something about type of dissolved organic carbon in the water. So you can say a lot about different things. All these papers are going to be written down there. I'm going to put them in Dropbox so you can pull them out. But just going to point out that a manual last year wrote a really, really good review of optical proxies that can be used to study particles in the ocean. And this was a fantastic book that was probably edited by Colin. Quite a lot of people from here contributed that one. It was a using optics in harmful algal blooms or something. And it was lots of chapters that you can find helpful when you think about optical proxies to study biogeochemistry. Okay, and then I think the coolest ones are the ones that allow us to study fluxes in the ocean. So, because the whole idea, I mean, it's great to know the quantity, but it's really cool to say something about changes of these quantities in the ocean. And when we talk about fluxes in general, I see them as a movement of quantity from one pole to another. And we can observe that as movement of that quantity from one space to another. So sedimentation, settling, or carbon export from the mixed layer to deeper ocean. So you can have a change, a flux of that property in time. And we can talk about the productivity. So primary productivity, you see the change of particular organic carbon, let's say over time. And then you have the flux that is change in type of property. So when you have transition of carbon to DOC, for example, I mentioned before those big photosynthesis blooms, when they start dying, most of the carbon, unfortunately, doesn't go to the export. It does go to DOC because all the goo that photosynthesis cells are sitting inside just goes in DOC. So we can quantify that using optics, or we can try. I haven't seen the paper, but it shouldn't be, it's doable, I'm sure. So I'm going to go slowly through each of these, some of these, and kind of lead you through it. This was a paper published by by Colin, and she is talking about what to use of all these available optical measurements to infer chlorophyll concentration. Why do we want to study chlorophyll biomass? I'm sure you guys heard a lot from Mary Jane. And here she's talking about, how to properly calculate this line height and use the line height, absorption line height in 676 to say something about concentration of chlorophyll. And here you can see that's a really good method. It really sits nicely. It is log scale, but it looks really nice. But she's also pointing out that this normalized absorption spectra, as you saw guys today, and you're going to see even more once you really clean up your data, that there's a large variability from species to species. And that for a single species, depending on the type of environment we keep her in, low light or high light, that spectra can be variable. So Thalassiosira, that little diatom that you guys ran yesterday, if you keep it in low light, this light blue spectra, it's quite different than the Thalassiosira that was kept in high light, this dark blue spectra. So that is a little bit of a variability that we can observe in this absorption to chlorophyll ratio. But it's much smaller than the variability that we see when we try to look at the chlorophyll fluorescence as a proxy for chlorophyll. Mary Jane mentioned a little bit about chlorophorescence. You're going to hear much more tomorrow. Chlorophorescence is one of the most abundant optical measurements in the ocean. I mean, everybody has a chlorophyrometer. Physicists, most of the physicists think that if they put a chlorophyrometer on their observation or whatever, they're doing biology. The same way, this is the most problematic, I consider it to be the most problematic optical measurement in the world, because everybody think, oh yeah, now I have an approximation of the biomass, but you don't. You have something that's a product of the biomass and the physiology of that cell in the phytoplankton community. So this is from a paper that we published recently, and you're going to find these kind of graphs all over the place, where you can look at the ratio of chlorophyll fluorescence and chlorophyll as a function of PAR. And there's always going to be the same pattern. At one point, this relationship is going to start dropping down. So you're going to start seeing less chlorophyll fluorescence over chlorophyll. And Mary Jane is going to go into detail, but that's happening because of the daytime quenching. There's quite a lot of light starts freaking out the way that the way the cell works, the way the chlorophyll functions. And that's going to be visible in chlorophyll fluorescence. And more in detail, Mary Jane is going to talk about. But we can see similar variability of chlorophyll fluorescence to chlorophyll ratio if you have algae and nutrient stress, or usually nutrient stress and different levels of nutrients. Second thing that is actually shown to be really the main driver of variability, you can see it in this graph that was also published by Colin. And here, once again, you see chlorophyll fluorescence over chlorophyll. And all these different bars are different species. So for any given species, we see that this ratio goes anywhere between 20 and 120. This is a gigantic diversity. So once again, if you want to use chlorophyll fluorescence as to say something about biomass, you can do it. It's doable. Here we managed to get from chlorophyll fluorescence to chlorophyll. It was a nonlinear function of PAR, temperature, depth, and time. So we did 4 levels of correction on it. And we use it only within the scales of our experiment. So we interpolated. And with some security, we can get this chlorophyll. But it's very promiscuous measurement. Just remember that. And never take this relationship that I've published and take it somewhere else, because it's highly variable. But Mary Jane's going to be talking about that tomorrow in details. Another cool measurement that everybody wants to make is the measurement of the carbon biomass. I think chlorophyll is awesome, but everybody in the end wants to get the carbon, just because that tells us a lot about, carbon fluxes in the ocean, just general biogeochemistry. As you heard a lot from Emmanuel, particles scatter, no? So we can assume that both suspended particulate matter and particular organic carbon are going to have some kind of relationship with backscattering and attenuation. And Emmanuel has shown quite a lot of these relationships. First time that attenuation was used to develop as a proxy for carbon was by Wolf Gardner, that Emmanuel mentioned before. It was first time in there was a Jagoff's cruise in North Atlantic, where he developed this relationship that allowed him to take the whole profile of the CP and calculate carbon from it, which is pretty awesome. So since then, quite a lot of people have went and measured this POC in conjunction with the CP, and they managed to get a good proxy for estimation of particular organic carbon. But you have to remember, because you guys now heard a lot about scattering, attenuation, that if we try to develop similar thing for backscattering, we know that backscattering, except for size and mass, is dependent on the morphology and type of the particle. And they can cause a little bit of a problem. When it comes to the biological particles, the carbon density varies a lot. You remember, I don't know from the last lecture how much you remember when I was talking about stuff, diatom is a gigantocell, no? So when you look at the diatoms, what is the most of that diatom cell? What is in there? Like, is it like nuclei? Is it the cytoplasm? What is the biggest thing inside of that cell? Well, there's like a word for it. There's a vacuole. It's a gigantic balloon filled with water sitting inside of that diatom. So if you would take a cross-sectional area of the diatom, and I may be like a little bit cartooning here, this is your diatom. This would be your vacuole, and everything else would be pushed here on the edges. So for the affected cross-sectional area, most of the stuff that has carbon is pushed toward the edges. So you would expect for the cross-sectional area you're going to have much less carbon than for a little flagellate cell that doesn't have that vacuole. So it's something to take with you in your head when you're trying to develop these things. And that's what I started facing when I was trying to develop these proxies for the North Atlantic bloom experiments. I took the particular organic carbon samples that we had collected and plotted them against backscattering here and against attenuation. And the red line is the one that I developed. But then I superimposed other smart people, because you really want to make sure that you're doing the good stuff, and everything was all over the place. I mean, for the attenuation kind of fitted, but when it came to backscattering, we had orders of magnitude differences. And now you start asking questions, is this because of the natural variability, or is it because of some kind of instrumental differences? They're quite widely possible. So I divided my data set in different groups, and what I saw that relationship between particular organic carbon and backscattering does not change as I switch from the diatom to flagellate community, recycling community. But as I go from the surface water that is dominated by phytoplankton to deeper water that is dominated by non-phytoplankton, so detritus and some mineral particles, their relationship changes statistically, dramatically. The story is completely different for particulate organic carbon and attenuation. As I go from diatom-dominated community, regardless where in a water column, to recycling community, so the teeny tiny guys dominated community, I see the change. And this change, this increase in carbon per CP, is actually this thing here. So Emmanuel was saying the CP is associated with the cross-sectional area of the particle, no? And I said here that diatoms have much less carbon per cross-sectional area than flagellates. So as I moved from diatom to recycling community, I saw that change. And that was kind of cool. So then I can explain a portion of the variability. And I can start plotting particulate organic carbon versus CP on a graph as a function of maximum CP. So this graph kind of similar to the one that Emmanuel showed from Hill, no? Where he was showing SPM versus CP and maximum SPM. And here, we're in the open ocean now, we're not in sediment. Maximum CP is actually a measurement of how much biomass do we have. The larger amount of biomass you have in the open ocean, there's a bigger chance that you're in diatom-dominated environment. And that's what is the base of quite a lot of these PFTs. If you guys are not following me, let me know so I can slow down. This kind of makes sense? What is the high? So high biomass in the ocean is always associated with this, like, it's always associated with diatom blooms. Just because that's at all these attenuation meters, C-star stuff like that always operate in red. So this is nominal 660, it's like 6 something else. But most of the oceanic measurements of CP are done in 660.
Let's think why.
Yeah.
Why do you think you would measure in the red if you want to do particles? What's the interfering to do in the group?
Mm-hmm. Exactly.
That's one reason.
And also, when CTEC first built the transformers,
The cheapest thing,
LEDs were available
only in red.
and not in any.
But to this day, those are the most stable ones.
What is that C76 for the chlorophyll?
Because that's your answer.
But why would you want it to be? If you're doing beam attenuation,
Yeah,
supposedly, and if the shape is relatively smooth, it
doesn't matter. And think about it, these beam attenuation meters and scattering, I think, and I can be corrected, were more used in sedimentology than in open ocean studying chlorophyll. So I really don't care because the sediments don't have, you're going to have an expression of chlorophyll peak rarely in really strong blooms in attenuation. Here, no. So, and I think it's 5 if you, and you guys can do that. You can take the radiometers and check out the wavelength of these of the meters, attenuation meters that you have. It's always nominal 660, but it's actually 653 or something. You can do it. It's really easy. Like we did with those LEDs. You guys can check. But regardless, this tells you that there's an increase in biomass, increase in size. And if you take my data and everybody else's data, well, most of the people's data, Wolf Gardner's data, we can all follow this kind of imaginative line, and this line is calculated using Wolf Gardner's data, who's much smarter, much older, knows much better than I do about these things. And kind of like everything's going down. So this trend is observable everywhere. The outliers and things going up and down, we assume are associated with a different technology, different instruments that people use in different ways they're measuring carbon, and that's going to come in play a lot. As Emmanuel was mentioning, were you talking about aperture, like the angle of the measurement of different meters? Yeah, acceptance angle, sorry. Have you spoke? Yeah. So I must have not paid attention, but they will also play important game in how much of the CP per carbon you're going to see. And it's very similar for the particulate matter. This is a paper from Griet. Okay, so you're going to, yeah. This is POC versus CP. So this is like POC CP slope. So this is your, it's pretty much POC over CP, yeah? Always. So most of the slides I'm going to be showing today are actually people who were associated with this class. They're either teaching the class or they took the class. Did Grit took the class? No, she never took the class. I think she's one. There's like two more slides, 2 more papers that are not associated with the people who took this class. Anyways, she did similar stuff like this. This is the measurements that were done, I think, in English Channel, where she's looking at the CP as measured by list and the backscattering measured by, I don't know, something, versus SPM. And she found that there's some trends, and the differences in CP to SPM, so suspended particulate matter in this case, are associated with the particle parent density. So how thick are they? And particle composition was the reason for variability in the backscattering to SPM relationship. It's a really cool paper. You guys should go and look at it. But and then there was that graph that Emmanuel showed just before, where it was kind of similar trends as we have seen similar trends. It was a similar graph as we have made. So Paul Hill is going to give a talk, I'm sure, about this, where he plotted CP over SPM versus maximum SPM. And so this kind of trend. So if you compare these two graphs, they're actually going in opposite direction. I'm looking at carbon over CP, and they're looking CP over SPM. And both of them going kind of this way. Do you guys know why? Because I'm not sure. I mean, like, I have theories, and I'm sure Manuel can help me. First going to help you to think, this is all happening mostly in open ocean. So if CP of 1, it's based on this one. CP of 1 is, let's do this. CP of 1 is give you 300 micrograms of carbon. Micrograms of carbon per meter cube. There's an error here. And that's actually not an error. This is maximum SPM grams per meter cube, 10 to the second grams, 10 to the third. This is a kilo of matter per meter cube. That's a lot. So we're talking about 2 completely different environments. Exactly. So this stuff is bottom boundary layer. What is driving this trend, as Emmanuel was saying, was dominated by big particles who are going to interfere to kind of mess up the measurement of CP. And what you see in CP is actually underestimating the amount of SPM that is actually present there.
I'm not understanding. So one's showing SPM versus the attenuation ratio with SPM. And
Yeah.
the other one's showing just
Particularly any carbon.
CP with the carbon slope.
It's pretty the same. It's the same. Forget about slope.
Over CP, the other is CP over mass.
Yeah. So just up.
This is
It-
carbon mass, this is weight.
Yeah.
But it's an inverse ratio.
It's an inverse ratio.
One could think, for example, just as an example of trying to explain it, when you have very low biomass in the ocean, what kind of cell dominates your sample?
What kind of particles dominate your sample?
Big or small cells?
Small.
So you can think of it maybe small cells on the
Yeah.
left-hand side.
This is small and this is bigger
submicron,
cells.
Submicron cell on the left.
There's no submicron cells.
Cyanos and cyanos and proteroplankton. Bacterial plankton.
Yeah. Small stuff, yeah.
Well, as you move to the right, you get more towards diatom dominance. And if you think about the resonance curve that we showed, you might move from the before you're getting into the resonance towards the resonance. So you're more effective in
Capturing the
just like on
CP.
size alone. You're more effective in scattering as you're moving to larger powder. As one explanation is what you're seeing.
Yeah.
Maybe.
But this is something that, like, I just want to point out, we don't have to find an explanation. Think about it. These are going to be papers that you're going to be encountering. That these are pretty much two same optical measurements that we use to infer something about particulates in the ocean. But they operate, they seem to act differently in 2 different environments. So just think about what are driving these opposite trends. We always want to measure this carbon, in particular organic carbon. What I really want to get to is phytoplankton carbon. Because particular organic carbon pool in the ocean is, the one that we talk about is usually comprised of particulate phytoplankton carbon and everything else. And everything else is detritus. So that in the side you have bacteria, junk, eaten some small zooplankton, things like that. And what we really want to know in order to be able to drive these prime productivity models and so on, we really want to say something about phytoplankton carbon. And up to now, up to recently, the way the phytoplankton carbon was calculated is that you would do some imaging of the phytoplankton. So either under the microscope or through flow cytometry, calculate the biovolume, and time it with some previously published values in cell-to-carbon ratio. And you would combine that with the flow cytometry and infer something about the small size, and that's how you would calculate the phytoplankton carbon. But just recently, a group from Oregon, and this was in a paper by Jason Graff, have started using cell sorter. So what they do flow cytometry, and they sort these cells. They accumulate cells of the same type, lots of them, in small cuvettes. And just cells. And then they take that sample and they run it for carbon. So since they know the number of the cells and the amount of carbon, they can say for that sample how much carbon was there in a single cell. But also since they measure the backscattering, they can make a really good relationship between backscattering and phytoplankton carbon. Why is this so important? Why do we want to connect these two? Why do we want to connect backscattering to phytoplankton carbon?
Isn't that what remote satellites are measuring?
Exactly. Backscattering is driving most of remote sensing. What we see from remote sensing, we can estimate backscattering really well from remote sensing. And if we know that, we can say something about phytoplankton carbon. If we know phytoplankton carbon, we can drive the prime productivity estimates better, and so on and so on and so on. So this is a really powerful measurement, really powerful method. First paper on it, we have to see what's going to happen. I can bitch about this paper for hours, but it's a fantastic, fantastic method and a great, great, great advance in our knowledge about the way we measure, the way we do carbon in the ocean. Okay, so I'm going to do one more slide. I'm going to stop and eat dinner. Okay. And then we're going to move in the dissolved area. So we can also do dissolved organic carbon pool. Here. I didn't say a thing about particulate inorganic carbon, which is an awesome, awesome, important pool of carbon in the ocean. And Jason will kill me if I don't say that. But his advisor is going to come over and give you guys a great talk on the 4th week about particulate organic carbon and how do they measure that one in the ocean, both in situ and from remote sensing. Dissolved organic carbon, another important pool of carbon. And really cool stuff, if you work in a coastal environment, you're going to, for the certain area, for the certain season, you're going to have a really nice relationship between dissolved organic carbon and ACDOM in your region. That's awesome, because then you can measure ACDOM, say something about dissolved organic carbon, you can measure the particulate carbon with backscattering, start talking about those transitions, those fluxes between the particulate organic carbon and dissolved organic carbon. However, when you're in the open ocean, everything falls apart. The sources of CDOM and DOC in the open ocean are completely different than the ones we find in a coastal area. The residence time of the dissolved organic carbon in the ocean is completely different than in a coastal area. So when you're in the surface, and that's 0 here, most of that CDOM actually gets bleached, photo bleached. When you're in a deep area, there's different processes that attack that DOC and CDOM in the ocean. But important thing that you can take home, that there's no relationship whatsoever between CDOM, absorption or fluorescence, and DOC. But if you're a coastal area, and if you know, again, if you're in a coastal area, you have these measurements done for your estuary, for your season, because the sources of the CDOM and DOC are different based on the season, you can have a really nice proxy to measure dissolved organic carbon in the water. So I'm going to stop here and just have some slides that I can do after lunch. So is this kind of more or less, I'm just going to tell you lots of stories and going through them. And just kind of to give you, open your minds to idea of different things that you can do with optics and how you can use it in biogeochemistry. And I'm sure you guys are going to be able to develop many, many more later on. Okay, food? Food. Okay, go.